Thank you, Professor. Uh, like to start my talk on keratoconus and its whole etiology and its what areas we have worked on. These are my financial interests. Historically, it's been known to be caused by eye rubbing. There's a genetic mechanism to it. And the most interesting aspect for us was this question mark about the unknown mechanisms. When you look at inflammation, the cardinal signs of inflammation, the heat, redness, swelling, pain, and loss of function, has been associated with inflammation. But when you look at these diseases like these, the question we need to ask is, does keratoconus have all these things, or it can come without any of these cardinal signs of inflammation? The first cardinal rule when we started was to not to discard anything, whether you are using an epithelium when you're doing cross-linking, the tears, whatever. Especially, these are all for the other parts of our research. The cardinal rule of any biological research is to preserve your samples. And more you preserve your samples, the more you keep your samples, the more pathways you uncover. And when you have the samples, it's important that you can go back and start analyzing them with your proteomics, your metabolomics, your gene pathways, and multiple other things. And that started this whole journey of looking at different types of inflammatory pathways. And many of these papers did suggest that there is a huge mechanism of inflammatory pathways and many altered other pathways which needs to be addressed and to looked at carefully. To summarize here, these are all the epithelial cell lines from the holotomography imaging, what we do on our corneas, and this is how an autophagy happens, where your corneal dead cells is eaten up by the neighboring cells. And when you create a dry eye or a keratoconic kind of model, you see that it's a hyperosmolarity status. The cells don't digest its neighboring cells. So what happens is these are one of the responsible for an autophagy, which is your housekeeping mechanism of your eyes. And when this happens, when you create a lot of inflammatory changes, this would have an effect on the collagen. And these effects on the collagen is seen as lowering of the collagen and the enzymes which holds the collagen together, the lysyl oxidase. And you can see that when you high level of MMPs would reduce the collagen and reduce the lysyl oxidase. This was one of the first experiment we did close to 11 to 12 years back. The second thing we started asking question is the microbiome on the ocular surface. It's a very challenging thing to look at the microbiome because we know that close to around 95-96% of the bacteria cannot be cultured. They are unculturable. So if you take up any sample, you'll get the same sample, staph and pseudomonas or commensals. So what we did was we tried to look at the genomic expression of the microbiomes. So we did the microgenomes expression, and then we correlated that with the immune dysregulation. So what was interesting in this paper, it's just a new paper which is just a few months back, there is the ones in red are the family of bacteria were completely different in keratoconic eyes compared to a normal eyes, and this correlated very well with all the immune dysregulation which we've been seeing. So two important things is, one, the role of the immune responses which is dysregulated. The second is the role of a microbiomes which is altered in keratoconus. Now, how do we understand, how do we take this forward? And this is something we worked on because we know there are a lot of cytokines and immune dysregulation, but we wanted to bring it to a point of care kit. And that's when we worked on this biomarker pathfinder, where from your tear film, we could detect a lot of major immune changes which is happening both in dry eye and your ocular surface. And this is a 90 minutes kit, 90 minutes test, and this is the immune cells. and you can study from your aqueous, vitreous, or any other samples what you given. So what you get is these immune cells, and they have different functions. They have functions in health and functions when they're not healthy. That means they can be altered functions like the immune dysregulation, fibrosis, inflammation, and all these factors. And then we build an AI on this. So now we have an AI. This is more for the dry eye, but also the same thing can be used for keratoconus. And the one in greens are healthier. The ones in red mean that they are completely very high. And it is both, it is quantifiable. That means you can quantify the amount of inflammation. Many times you have these MMP kits and other things which tells you yes or no. Yes or no means it may be here or it may be here, and no means it may be just at the edge of green. But here it is positive, but it also tells you in what level. And this went through close to around 5,000 dry eye samples and more than 3,000 keratoconic samples to create these pathways. It's important to understand the immune profiling because the failure and allergy also has an impact on the chance of the cross-linking outcomes. To study the whole collagen, we build our own indigenous polarization OCT. The polarization OCT is where we use, instead of a single unpolarized light with multiple wavelengths, we use a polarizing filter which uses one polarized light, and then it gives you the birefringence properties of your collagen. So when you're looking at the stable and a progression keratoconus, you start looking at multiple things. One thing we started seeing is the Bauman's layer. When you have a progression, the Bauman's layer is much thinner compared to a stable keratoconus. So Bauman's layer in keratoconus becomes a very important structure, both from an imaging point of view and also from a biological aspect. Because what we believe is when you have a thinner Bauman's layer, your inflammatory molecules can pass through your stroma. And this is what we see. A thinner Bauman's on a polarization OCT has a high level of dysregulated immune profiling and a more aggressive progression. So this helped us to create this model where you're looking at an eye environmental insults, atopy, allergies, cytokines, change in the collagen. Both it has a feedback forward loop and the feed loops, which changes the whole collagen structures. So we are looking at structural collagen, the Bauman's layer, the inflammation, the bacteria, and all these factors drive you to go into a progression. Now comes the second part of the work. When you know that you have identified strong evidence of inflammation, identified there are changes happening, now can we use medical way or some other way to stabilize them? And this paper we published with, this was one of my first papers on this subject. We used the cyclosporin because cyclosporin reduces TNF alpha, and this TNF alpha reduces the change in your, it reduces the degradation of your collagen. And we found that it improved the status of the collagen health. Then came the second thing where I showed you the image of a cell eating the cell, then autophagy mechanism, and what we found was, this is a schematic we use, that it works on the TGF beta pathways, it works on the collagen extracellular matrix. It also has an impact on the fibrosis. So trehalose improves autophagy, it improves the collagen synthesis, it improves the lysyl oxidase. And there's also a role for lifitegrast because lifitegrast also has a role in that at the T cell inflammation. And what was interesting here is this is the lysyl oxidase and which holds the collagen. There's a nuclei, and in the keratoconus we can see that the levels are. This is what in the control, and in keratoconus it's already gone down because we induced TNF alpha onto this so that the collagen and lysyl oxidase is lower. So when you use trehalose, which improves the inflammation, the lysyl oxidase, in spite of inflammation, it improves the collagen synthesis. That's the beauty of biology, is that you can translate what you see in your clinics from your lab. You can, and as a surgeon, you can bring it to your clinics very quickly. And then we understand there is a basal load of inflammation which causes multiple changes. And when you do a cross linking, it's important that your haze is driven by the level of inflammation, what it is. So it's always important for us that you give a small amount of biological cleansing, and this work was presented by Dr. Deepthi in the Congress, the TNF alpha and haze. That means this is TNF alpha before we do the procedures, which I can pick it up from my biomarker kit. That higher level of TNF alpha causes more haze, more excessive flattening. So what we can do is we can cleanse them biologically by giving cyclosporin, lifitegrast for 3 months before we do any cross-linking, and you can reduce the amount of haze completely. So this is what I believe. I call it as a biological cleansing because your basal inflammatory threshold are responsible for the outcome of any procedures. And I was just discussing with Dr. Samar Basak sir that it also has a role in your endothelial cells and endothelial grafts. Now, moving on to genetics, we started looking at the genetics and gene panels. We worked on new genes which may have an impact on the on early detection. We started focusing on lysyl oxidase because that is what helps the collagen to strengthen. We started looking at whether we could do a point of care kit which could be faster. We failed in identifying from the tears, but we are still able to do something in the tissues. We then looked at this is a genetic of keratoconus. Even if you don't have a keratoconus, the sisters on collagen still shows like they have a keratoconic picture. And then we started looking at one of the areas of work was to start looking at the gene augmentation of lysyl oxidase. This is our facilities where we make our own vectors and started working on it. And this is just a picture showing that one injection on the rabbit model is able to show very good response. We are going to start, if everything goes well, the first gene therapy trial by end of this year in keratoconus. We make an intact channel and we inject into this. So there is inflammation, there are different levels of medical lines of treatment, and I think genetics and gene therapy has a huge future. Thank you.
Thank you very much, Rohit, for this wonderful talk.